Good morning, good afternoon, or good evening, everyone. This is Biology 205 at Illinois Central College, and today we're going to be talking about the nervous system and covering Chapter 12 in our text. The learning outcomes for today's lecture include the following: to describe the anatomical and functional divisions of the nervous system for both the central nervous system and the peripheral nervous system, to describe the anatomical and functional divisions of the peripheral nervous system, including both afferent and efferent divisions, to describe the anatomical and functional divisions of the afferent nervous system, and that's a specific division of the peripheral nervous system, to also describe the functional divisions of the autonomic nervous system. You'll also explain the structure and function of a typical neuron and its coverings, and review the organelles of the neuron and recognize what traditional organelles are missing, and discuss the significance of not having that particular organelle, to classify types of neurons based upon their structure as well as their function, to list the pathway of neuron organization, the input to processing to output. You'll compare different classes of sensory receptors, and that includes somatic versus visceral sensory, and interoceptors versus exteroceptors versus proprioceptors. We'll also describe the main functions of interneurons and discuss the structure and functions of different types of neuroglia. Also, we'll compare the characteristics of a myelin sheath and how it differs between the central nervous system and in the peripheral nervous system. We'll contrast an internode to a node of Ranvier. We'll compare white and gray matter and associate that with structural regions on neurons, and describe what a transmembrane or resting potential is and how it's maintained. We'll explain what an electrochemical gradient is and how it relates to the action potential. We will explain the function of the sodium-potassium ATPase, which is adenosine triphosphatase, through the sodium-potassium pump. We will also compare the neuron membrane channels, how they're controlled, and what moves across the membrane. We'll explain how gated channels are regulated. This includes voltage, ligand, and mechanical mechanisms. And we will explain how the resting membrane potential is created and maintained, including the electrochemical gradient, membrane ion channels being both gated and non-gated. Also, we'll describe and understand the events involved in the generation of graded action potentials and actual action potentials, including the activities at the axon hillock, the initial segment, which is also known as the trigger zone. And then we're going to look at local/graded potential establishment. And this includes temporal summation and spatial summation. We'll also explain the role and significance of calcium at the presynaptic terminal. We'll utilize the following terms properly. That includes depolarization, repolarization, hyperpolarization, threshold, and all or none. We'll also draw an action potential, label it with the terms that we've used above, and describe the movement of ions and the regulation of those channels. We'll differentiate between an absolute and the relative refractory periods. We'll explain the difference between continuous and saltatory conduction. We will contrast types A, B, and C axons and describe how that affects the speed of propagation. And we'll explain what an electrical synapse is versus a chemical synapse and where they would be found. Last list of learning outcomes. We will describe and understand the general structure and function of chemical synapses in the peripheral nervous system as well as in the central nervous system. We'll list the function of a chemical synapse and describe the structural parts. We will identify what a neurotransmitter is and how it's used at a synapse. We'll describe the process of removing a neurotransmitter, and this includes reuptake or reabsorption, enzymatic degradation, and dilution. We'll describe the events that occur at a chemical synapse and understand their significance. We'll define synaptic delay as well as synaptic fatigue. We'll also describe and understand the function of the major types of neurotransmitters and their basic effect on the postsynaptic membrane. This includes acetylcholine, norepinephrine, dopamine, and serotonin. We will explain the function of a neuromodulator and provide an example of how it works. So we'll discuss GABA and opiates. And we will also explain the interactions that occur during the processing of information in neural tissue. So this includes IPSPs, EPSPs, summation, inhibition, and facilitation. I know it sounds like a lot. Let's get going. In today's lecture, we're going to follow the outline of the text again. So let's start with section 12-1, the nervous system structure and function. So overall, when we discuss the nervous system, we're going to include various organs in this discussion. Primarily, we're thinking about the brain and spinal cord, the receptors of sense organs, such as the eyes and ears, etc., and the nerves that are going to connect to other systems. But we're going to break all of this down into sub-constituent parts coming up during this lecture. So when we discuss nervous tissue, we're really going to be discussing two primary general types of cells, and that will be neurons, which is the primary cell that communicates with one another, and neuroglia, which are also known as glial cells. And these are the support cells that support the neurons, so that way the neurons can do the communicating. So these neuroglia are going to be essential for the survival of the neurons. They also help maintain the structure and the function of neurons. So the neuroglia are absolutely essential. So let's discuss a little bit in further detail the anatomic divisions of the nervous system. We'll discuss the central nervous system and the peripheral nervous system next. The central nervous system is what you usually think of when you think of the nervous system, because this is the brain and the spinal cord only. So within the brain and the spinal cord, this will include nervous tissue, but also there's connective tissue and blood vessels present within the central nervous system to support the brain and spinal cord. So this will function to process and coordinate all the sensory data that we're gaining from our environment, both external and internal of our body. This will also control our motor commands, so that way we control all the activities of our peripheral organs, like our skeletal muscles, so that we can run and jump and sit down. And it will also be responsible for controlling higher functions of the brain, which include things like intelligence and memory, learning, and emotion. The peripheral nervous system does not include the brain and spinal cord, but basically it includes everything else that isn't the brain or the spinal cord. So that means it's all nervous tissue that is outside of the central nervous system and the enteric nervous system. So this will deliver sensory information to the central nervous system so that the central nervous system can process it. And it will also carry motor commands to peripheral tissues on our behalf. So nerves, which we think of as being within our limbs and appendicular skeleton, are actually throughout our entire body. But these nerves are really just bundles of axons that have connective tissue and blood vessels around them to support them. And they'll be carrying sensory information and motor commands. So you can basically divide everything that a nerve carries into either it's sensing something or it's sending a motor command. It's one of those two things only. So then there are different types of nerves, and those that attach to the brain directly are going to be called cranial nerves, and those which attach to the spine, specifically the spinal cord, those will be called spinal nerves, and we'll discuss those later in further chapters. The peripheral nervous system is broken down to two different functional divisions, the afferent division and the efferent or efferent division. The afferent division is going to be carrying sensory information, and it takes it from the receptors that are in our peripheral tissues and in our organs and brings that information up to the central nervous system, which is the spinal cord in the brain, so it can be processed. Now, the efferent division is more of a business division because this is actually carrying the motor commands to tell the boots on the ground what they need to do. So efferent signaling will bring signals from the central nervous system, again, that's our brain and our spinal cord, and it will carry those motor commands out to our muscles so we can move, and out to our glands so they can secrete their product, and also to adipose tissue. The peripheral nervous system is also further divided into its functional divisions, so how it works. So we'll break it down into either receptors or effectors. So receptors are anything that detects whatever that change might be in the environment to respond to the stimuli. And this can either be in the form of being a specific neuron or a specialized cell to complete this task. And this can also be either a single cell just operating on its own being a sensor, or it can be many different cells working in tandem together, such as a complex sensory organ like our eyes or our ears. Also, we have effectors. Now, effectors are going to be the target organs that actually respond to the motor commands. So they're responding to the information that comes from the central nervous system. Before we go any further, I just want to share with you a quick way to remember the difference between afferent and efferent. So the way that I've always thought of it is using the word SAME as an acronym for each of the words that I need to put together to recall. So SAME stands for sensory afferent, motor efferent. So hopefully that will help you a little bit when you're trying to keep this straight. So when we discuss the efferent division of the peripheral nervous system, we're going to break it down into either the somatic nervous system or the autonomic nervous system. So the somatic nervous system is going to be controlling things that are both voluntary and involuntary, but it will be the skeletal muscle being able to contract. So when I say that this is both voluntary and involuntary, what I'm saying is voluntary movement is something like picking up your cup of coffee to have a drink. Involuntary, on the other hand, is, for example, if you put your hand on a hot element and you immediately retract your hand before you even have a chance to think about it. And that's because there's a very specific reflex path, which we'll discuss again coming up in future lectures in more detail, but that's an involuntary skeletal muscle contraction of the somatic nervous system. Now, the autonomic nervous system is totally out of our control, and we can't do anything about this at all. So it's controlling all of our subconscious actions, like the contractions of smooth and cardiac muscle and glandular secretions. We break this down further into two categories, either the sympathetic division or the parasympathetic division, and we'll be discussing this also more in future lectures. But for today, the general idea is that the sympathetic division is stimulating. So this is the fight or flight response. And the parasympathetic division has more of a relaxing effect. So we call the parasympathetic division the rest and digest division. The enteric nervous system, or ENS, is composed of 100 million neurons plus in the walls of the digestive tract. And there can be as many or more than that in the spinal cord. These use the same neurotransmitters as the brain do to coordinate information like visceral reflexes, and so that will initiate them and continue them. And it does this without instructions from the CNS. So the enteric nervous system is somewhat autonomous, and it can be influenced, though, by the autonomic nervous system as well. Figure 12-1 in your text gives you a general overview of how all of these parts come together to allow us to respond to our environment. So let's start on the bottom left, down with all the receptors by the feet of this individual, standing in anatomic position, keep in mind, with the palms facing anterior. So what we're looking at the very bottom we have special sensory receptors, visceral sensory receptors, and somatic sensory receptors. And all of these receptors are going to pass information through the peripheral nervous system, through the afferent division, remember sensory is afferent, up through the afferent division, up into the central nervous system, which includes the brain and the spinal cord. At that point, our brain can process the information, integrate it, and coordinate all of these different pieces of information so that way it can send a motor command down through the efferent division. And that will be divided into either the somatic nervous system if it's going out to our skeletal muscle, or the autonomic nervous system if it's going out to any of our other parts of our body, such as smooth muscle, cardiac muscle, etc., etc. So those autonomic nervous system messages will either be through the parasympathetic division to rest and digest, or messages through the sympathetic division telling you it's time for fight or flight, so get busy and get active. Let's move into section 12-2, neurons. In this section, we're going to discuss how neurons, being the functional unit of the nervous system, are intercellular communicators. Neurons are the basic functional units of the nervous system. They're responsible for sending and receiving signals that coordinate communication, information processing, and control. So we're going to look at some images in a moment showing you the overall anatomy of an average neuron. And the first thing you're going to look at is the cell body, also known as the soma. And this is the largest component of the cell. And it includes in it a very large nucleus and nucleolus, which is quite prominent, a perikaryon. And so peri meaning around and kary being nucleus. So this is the area around the nucleus. There's also mitochondria in this cell, of course, because it needs to produce energy. And we have rough endoplasmic reticulum and ribosomes, which are prominent because these synthesize proteins and have a very important role. Like all cells, neurons have a cytoskeleton of perikaryon, which is the area around the nucleus, and in the cytoskeleton there are a number of structures which help to hold the cell in place so it doesn't just collapse upon itself. So some of the things that you'll find in the cytoskeleton are the neurofilaments and neurotubules, and these are basically just similar to the intermediate filaments and microtubules we've discussed before. There are also neurofibrils, and these are bundles of neurofilaments, and they also help to provide support for the dendrites and the axon. So let's just look at the image down below for a quick second. So figure 12-2 in your text shows you a standard multipolar neuron. And what you'll see is at one end, at the left, you're going to see a whole lot of little receptors, and those are all dendrites. sticking off of the main perikaryon. And then we also have one extremely long structure, and that's the axon. So the way I'd like you to think of this is that the dendrites are receiving all the information, and then they are going to send the information out singularly through the axon, moving to the right. Also, we're going to have something called Nissl bodies, and what these are just dense areas of rough endoplasmic reticulum and ribosomes that are in the perikaryon. So all of these Nissl bodies basically make the tissue appear gray, and this is why we call the tissue gray matter versus white matter. The gray matter has a lot of these cell bodies. The dendrites that you see sticking off of the left side of the image below are all a bunch of short and very branched processes that are going to extend from the cell body, and they're all going to receive information from other neurons. They're very fine and they're very small, but because they're very fine and small, you'll notice that they also have a branching pattern, which gives a little more surface area, not so much exactly for absorption, but so that there are more contact points with other neurons, so they can receive an awful lot of information. And so all of these dendrites cumulatively will take up about 80 to 90% of the neuron's surface area. So the longest part of the neuron that we're looking at is shown in red below, and that's the axon. And the axon is just a single long cytoplasmic process, and what it does is it will propagate an electrical signal, which is an action potential, and we're going to discuss this more coming up. But it's going to carry that electrical impulse from the cell body on the left through the axon to the right and out towards the telodendria on the right, so that way the action potential can communicate with the next cell, which is not seen in this image. The axoplasm is just another name for what is inside the axon. So that is the cytoplasm of the axon. And inside of it, it's going to contain neurofibrils, the neurotubules, enzymes, and various organelles. Other structures of the neuron include the axolemma, which is just the plasma membrane around the axon, as opposed to being the plasma membrane around the cell body. So it's the axolemma. The initial segment, as you'll see if you look closely at the image towards the left, you'll see is the very base of the axon. So that's the very first part of the axon. And then we have something called the axon hillock. And the axon hillock or hillock is a thick region that attaches the initial segment, which is the first part of the axon, to the main cell body on the left. Additional structures on the axon to be familiar with include collaterals, which are branches off of the main axon. And there are also telodendria, and the telodendria are going to be those fine extensions on the most distal part of the axon. If you look closely at the image on the right, you'll see what's labeled as a postsynaptic cell, and that's the purple cell on the right. And so the telodendria are going to be trying to communicate information with that postsynaptic cell. There are also axon terminals, also known as synaptic terminals, and these are the very, very tips of the telodendria. Axonal transport, or axoplasmic transport, is just the movement of materials inside the cell moving from left to right in the image before that we looked at. So it would be moving from the cell body through the long axon out towards the axon terminal so that it can communicate with other different cells. This is called anterograde. But if there are waste products, let's say, from a number of different chemical reactions, then those products may need to move backwards, back towards the main body of the cell for processing and breakdown. So we call that retrograde flow. So the anterograde flow is going to be carried by a protein called kinesin, and the retrograde flow is going to be brought back by another protein called dynein. When we look at images of neurons in textbooks, we usually look at sort of a standard looking neuron. So every image that we've looked at so far is what is called a multipolar neuron. But there are actually 4 different types of neurons based upon the way they are structurally presented. So we're going to go through all 4 of these now. So first, we'll look at anaxonic neurons. So that's A in the image to the right. And anaxonic neurons are really small, and you'll see that all of those cell processes sticking out of them all look similar. So there's a whole bunch of what looks like little dendrites, but you don't see any specific long axon. Hence, they're anaxonic. They don't have an axon. So that's an easy way to remember these, and they're going to be found in the brain and in various special sense organs. If we move over to the image on the right, you'll see a bipolar neuron, which is really interesting because we have the cell body right in the very dead center of 2 long dendrites extending out but one is a dendrite and one is an axon. So one is going to be receiving information from all of those dendritic branches. It brings the information into the cell body, and then the information is going to travel down through the axon out towards the axon terminals. So we call this a bipolar neuron because the two processes are going to be separated with the cell body in the center. And we'll find these in our various special sense organs like sight, smell, and hearing. Unipolar neurons have one big long axon, okay? So the axon and dendrites are fused in this situation. So you can see up at the very top of the image on the left, you have many small dendrites that connect via an initial segment to the axon, and the cell body is just sort of popped off over to the side along the length of the axon. So this is a little bit different, but this is what is present in most of the sensory neurons of our peripheral nervous system. Multipolar neurons are what we consider our standard classic anatomy of a neuron. But these are a little bit different too because they have dendrites that are all stretched out at one end over the cell body up at the top of the image, and then a single long axon, which in this image is going down towards the axon terminals. So this is common in the central nervous system, and all of the motor neurons that control our skeletal muscles are multipolar neurons. So now that we've discussed the anatomy and the structure of different types of neurons, we're going to talk about the physiology and function of the different types of neurons. So we're going to break down neurons into categories of either being sensory neurons, motor neurons, or interneurons. So sensory neurons, which are afferent neurons, remember that acronym SAME, sensory afferent, motor is efferent. These are unipolar neurons, and their cell bodies are going to be grouped up into what is called sensory ganglia. So the processes of these neurons, which are afferent fibers, are going to extend from the sensory receptors up to the central nervous system, which is our brain and our spinal cord. So we have breaking down sensory neurons into either somatic or visceral. So we have sensation both in our external environment, so somatic sensory neurons can be expected to be found in our skin, and visceral sensory neurons are things that monitor the internal environment. So for example, things that monitor the sense of how full our bladder is or how full our stomach is. Those are visceral sensory neurons. Sensory neurons are responsible for carrying sensory information, but they are not the receptors that actually initially receive that information. They're the carriers. So we have sensory receptors that are responsible for actually sensing different things. So we'll break these down to three categories: interoceptors, exteroceptors, and proprioceptors. So interoceptors and exteroceptors are exactly what you think. Interoceptors monitor the internal systems of our body, like our digestive system, our urinary system, and also internal senses such as stretch or deep pressure or pain. Exteroceptors are going to monitor our external environment, so it'll sense things like temperature. And it also is important in complex senses like sight, smell, and hearing because they do help us to understand and adjust to our external environment. Now, proprioceptors are different in that they will monitor our position in space and time and the movement of our skeletal muscles and joints. So the way that I've always thought of proprioceptors is if you ever watch LeBron James or, select some famous athlete of your choosing, and you'll notice that these really amazing athletes have a fantastic ability to know exactly where the ball is in space and time. So when they dribble a ball in between their legs, they do it without even looking at their hands because they know if they move their hand a certain way, the ball is going to bounce exactly where they want it to and to come up again on the other side. That is having excellent proprioception skill to be able to monitor their own personal position and movement in space and time, in addition to monitoring something else like a ball. So proprioception is really important in athletics. Now, motor neurons, these are the efferent neurons. So remember, sensory afferent, motor efferent. The motor neurons are going to carry information from our central nervous system, the brain and the spinal cord, out towards the periphery effectors so that something can happen. So they'll be carried by efferent fibers, right? Because they're efferent neurons. So the efferent fibers have axons that carry this information. And we'll break this down into two categories, either somatic motor neurons or visceral motor neurons. So the somatic motor neurons are going to innervate skeletal muscles, whereas the visceral motor neurons are going to innervate pretty much everything else, all of the other peripheral effectors. So that includes everything from smooth muscle to cardiac muscle, our glands, and adipose tissue. When motor neurons are carrying a signal, they're carrying the signal from the central nervous system, so from the brain and spinal cord, and they're sending it out towards visceral effectors in our body. And when it does so, it's going to cross a ganglia. And the ganglia is going to divide the axons into two different groups, either preganglionic fibers or postganglionic fibers. And really all this means is it before the ganglion or is it after the ganglion? That's it. Inner neurons are the liaisons of the central nervous system, if you want to think of it that way. So they will be mostly in the brain and spinal cord, and you'll find some in autonomic ganglia. And what they do is they're positioned immediately between a sensory and a motor neuron. So this is going to be very important when we discuss reflexes later on. But they're basically that one part of the process that's responsible for being able to both distribute all the sensory information to where it needs to go and then to coordinate motor activity so that way we can respond to our environment. This is also involved in higher functions like in our memory, when we're planning for the future or trying to learn information like the nervous system today. Now let's move into section 12-3, neuroglia. And we're going to discuss how neuroglia are the support cells that help to support and protect neurons so that the neurons can go about their business of communicating. Neuroglia are the cells that are going to support and protect the neurons. They're like your BFF or your wingman, basically. So they're going to make up half of the volume of the nervous system, and they're found in many types, both in the central nervous system and in the peripheral nervous system. So as you can see below, we have figure 12-4, which shows you a breakdown of all the different types of neuroglia that we can expect to find in our nervous system. And you can see that there are 4 types that are present in the central nervous system and 2 types that are in the peripheral nervous system. So the central nervous system will have astrocytes, ependymal cells, oligodendrocytes, and microglia, whereas the peripheral nervous system is going to have satellite cells and Schwann cells. We'll discuss these all further. The neuroglia that we'll find in the central nervous system are astrocytes, ependymal cells, oligodendrocytes, and microglia. So let's look at astrocytes first. So astrocytes play a role in maintaining the blood-brain barrier, which is a selectively permeable membrane that separates the brain from all of the other contents of our body that could be present, for example, in our bloodstream. So there could be things in our bloodstream which may be toxic to our brain, so that semi-permeable membrane will allow some things to pass, like various ions and nutrients and dissolved gases, but it also is going to be important to absorb and recycle various neurotransmitters. It's also an important structure because astrocytes are going to form scar tissue after an injury has occurred. Now, ependymal cells are responsible for creating cerebrospinal fluid, which is the fluid that creates a kind of a cushion around our brain so we have a little bit more buffer against any potential injury. So the cerebrospinal fluid is going to be both created by the ependymal cells, but it's also going to circulate it via ciliary extensions. And the ependymal cells monitor the quality and volume of the ongoing cerebrospinal fluid. So it's a self-monitoring system to determine whether or not it needs to make more CSF or whether it needs to maybe add more water to it if it becomes too concentrated. So it's constantly making and monitoring and circulating cerebrospinal fluid. Now, this is something that you're going to see clinically. Sometimes you'll see somebody go in for a cerebrospinal fluid tap. A spinal tap is usually what's called very shortly. And so what happens there is they'll inject a needle into the fluid, draw it out, and then that can be tested for things like meningitis. Now, oligodendrocytes are there to myelinate the axons of the central nervous system. And myelination is a really important feature because it speeds up the passage of an electrical impulse from neuron to neuron. So the oligodendrocytes just speed things up basically, and they also create a structural framework as we're going to look at in the next image. Microglia, lastly, are there to basically eat up any dirt, damage, old things. They're just going to be the vacuum cleaners of the central nervous system, cleaning up all cellular debris. This is figure 12-5 in your text, and I absolutely love this image because it shows you all of the different microglial cells that are in the central nervous system. So starting from the top, you see on the right the ependymal cells. So those are the ones that are having a simple cuboidal epithelium, and they line the fluid-filled passageways that are both in the brain and the spinal cord. So you can see that there they are lining the central canal right up at the very top right. We also have some microglial cells moving just down one. And so those are phagocytes eating up debris and anything that shouldn't be there. So they're just the vacuum cleaners. We have the oligodendrocytes. And so you can see that the oligodendrocytes wrap their extensions around the surfaces of axons of various neurons. And in so doing, it creates a faster surface to allow a greater transport of the action potential in our processing. And then lastly, at the very bottom, we have an astrocyte, and the astrocytes have various attachments that attach to capillaries. And what they do is they create that blood-brain barrier that we just talked about. So all of those extensions anchor down capillaries so that they can filter out the fluid that is in the bloodstream to determine what gets into the brain and what doesn't. In the peripheral nervous system, there are only two types of neuralia, satellite cells and Schwann cells. So the satellite cells have a role in surrounding the neuron cell bodies and ganglia. So they'll regulate things coming and going into the cell, such as oxygen, carbon dioxide, nutrients, and also various neurotransmitter levels. So they're going to be important for regulating the interstitial fluid around the neurons. They're constantly communicating in that way. Now, Schwann cells, also known as neurolemocytes, are going to form a myelin sheath around various axons. And when they do this, they're speeding up the process by which an impulse can travel along that axon. So the very most outer surface of a Schwann cell is called the neurolemma, and it folds over kind of like a jelly roll. I'll show you in a minute. And it's going to have one cell. So a myelating Schwann cell will sheath only one axon, but many Schwann cells will sheath an entire axon. So you'll see lined up on one axon a whole bunch of Schwann cells lined up in a row, so that way they myelinate the whole entire length of the axon. Figure 12-6 in your text shows you a standard multipolar neuron with all of those little Schwann cells lined up one by one all along the entire length of the axon. And you can see that it's wrapped around kind of like a jelly roll. So it's separating that axon from the external environment and insulating that axon to facilitate the speed by which an action potential can travel along the axon. So you can see the blue axon extending from the top. So we've got the cell body with the nucleus in it and the perikaryon holding various organelles. Then we move into the axon hillock, and then moving down into the initial segment of the unmyelinated axon, which is the blue. And so you can see all of those tan sections, and those are considered myelinated internodes because they are myelinated by the Schwann cells. But you can see that there's a little bit of a space in between each of those, and those are called nodes or nodes of Ranvier. So let's take a minute to look at this image, and let's look really closely at it. So on the left, you're going to see what looks like a single cell, which it is. It's a Schwann cell, and the largest component of it is the nucleus. And you're going to see two cytoplasmic extensions that look like arms reaching out to phagocytose some structures within. And those are just the cytoplasmic extensions of the Schwann cell. And then we have the axons, which are all those little blue marks on the inside. So we have multiple axons of multiple neurons. So we have actually, I think, 9 different axons inside the arms of that Schwann cell. So you'll note in the image below then to the right that there are only 3 Schwann cells on that neuron, or so it appears, but you'll see that there are more extensions up at the top. So you see all of those axons going into Schwann cell number 1. So there's several in there. Schwann cell number 2, you can see just transparently that there happen to be multiple axonal tracts running through the Schwann cell. But then when you get down to the very bottom of the image, you'll see in the very dead center that there is the nucleus of the Schwann cell, just like that image at the top. And then you can see how it's wrapped around all of those different axons. So a single Schwann cell can wrap around multiple axons together, creating a bundle. Figure 12-6 also shows you a transmission electron microscopy image zoomed in at 27,000 times to be able to show you that there is a single Schwann cell myelinating a whole number of unmyelinated axons of multiple neurons. So you'll see that there are multiple Schwann cells lined up along the axonal length, so that way it is able to insulate and myelinate the entire length of the axon. Earlier, I'd mentioned that Schwann cells create something like a jelly roll around the axon, and this is what I'm talking about here. So if you look in step 1, you'll see that the Schwann cell is wrapping around a single blue axon, and it's just starting to encircle it. At step 2, the Schwann cell has completely encircled that axon, and now it's wrapping around it so it's completely enclosed. But at step 3, you'll see that the Schwann cell rotates, and so the myelin gets wound around and around and around the axon, like you're rolling up string, basically. So it creates a really tightly packed membrane, so that axon is completely myelinated and completely secure. While neurons have a difficult time repairing themselves in the central nervous system, we do have some ability to have nerves regenerate in the peripheral nervous system. So the neural response to injury that we're going to discuss today is called Wallerian degeneration. And Wallerian degeneration is really kind of a neat process. So if you look at the image at the top, you'll see that we have a standard multipolar neuron that has been transected at a certain point, and we're calling this the site of injury. So we want to be able to have that axon reconnect so that way we can retain normal function. And the way that that happens is we actually have the Schwann cells which will replicate and proliferate and grow together. So they kind of create the framework by which the axon then, once the Schwann cell is in place, it grows through the center of that myelination so that way it can reconnect. But it's not driven by the axon itself. The actual structure of the Schwann cell has to be there intact, creating that attachment first before the axon can actually grow through it. Now, nerve regeneration, like I mentioned earlier, is limited in the central nervous system because we have astrocytes, and those astrocytes will produce scar tissue, and they also release chemicals that are going to block regrowth. So Wallerian degeneration is something we will expect to see in the peripheral nervous system. Now let's discuss section 12-4, which is membrane potential. In this area, we're going to discuss ion concentration and membrane permeability. Earlier in the course, we discussed how all cells have plasma membranes, which is the outer surface of them, and that those plasma membranes are going to be selectively permeable, meaning that they allow some things to pass and others not. And when we talk about ions passing back and forth between that plasma membrane, this is particularly important to neurons because neurons are going to be passing electrical impulses in the form of an action potential. So the relationship between that neuron and the extracellular environment is extremely important. So the membrane potential of a resting cell is called the resting membrane potential. And if there is just a temporary localized change in the resting potential, we'll call that a graded potential. So this is caused by some stimulus, in which case there is now a difference in the electrical environment, but nothing is actually technically happening in terms of an action potential. An action potential is an electrical impulse that is first started by that graded potential, which is the beginning of the change. But the action potential is the full transmission of an electrical signal along the entire neuron to another neuron. The main take-home points about the resting membrane potential is really just that there is a difference between what is outside the cell and what is inside the cell. So the extracellular fluid and the intracellular fluid, or cytosol, are going to have different ionic compositions. So the extracellular fluid typically has high concentrations of both sodium and chloride, whereas the cytosol, the fluid inside the cell, is going to have high concentrations of potassium and negatively charged proteins. So that semi-permeable membrane is going to allow these sodium, chloride, and potassium ions to pass back and forth depending on what the cell needs. And also the membrane permeability is going to vary by ion. So it's not going to just increase sodium and chloride and potassium all at once. It's either going to increase sodium or decrease potassium or vice versa, but it's not just up or down, yes or no, everything moves in or everything moves out. We're going to have to keep this environment regulated very carefully, and that is the role of the selectively permeable membrane. There are passive processes that act across the cell membrane, which will allow for change between that plasma membrane, and one is called the current. And the current is just the movement of charges to eliminate a potential difference. So this means that positive charges are going to be attracted to negative charges, and they're going to move together to eliminate the difference between them. So that is what a current is. Now resistance is kind of exactly the opposite. It's how much the membrane will restrict ion movement so there is less current and less flow. So if the resistance is high, we will see a very small or slow current. Figure 12-8 in your text is a spotlight image, and it shows you what the processes are that produce the resting membrane potential. So all of our cells have a membrane potential because there's a difference in the energy on one side of the cell versus the other, the interior or the exterior. So that potential difference there is going to be called the resting membrane potential. So there are both passive and active forces that will affect this. So here are some passive things that happen. So first off, we have a passive chemical gradient. So basically what that means is if there's higher, let's say, sodium on one side and lower sodium on the other, we'll have a variety of mechanisms that will leak things through. So for example, we have potassium leak channels that will allow potassium to leak in and out. Now that's a passive process. We also have active processes like the sodium-potassium pump we've discussed before. So when the sodium-potassium pump is exchanging sodium for potassium, it's actually taking ATP and converting it into ADP, so that way it can have the energy to create that pumping action. In the previous slide, we looked at passive chemical gradients, showing that there could be either more, let's say, potassium on one side of the plasma membrane and less on the other, and that creates a gradient. But we also have an electrical gradient in which we have either a negative charge on one side or a more positive charge on the other, and that's also a form of potential energy. So the resting membrane potential is going to be measured in terms of millivolts, and the resting membrane potential for most neurons is about negative 70 millivolts. So start committing that to memory now, because this is going to come up over and over in the next several slides. So that negative sign is showing us that the inside of the plasma membrane is negatively charged as compared to the exterior of the plasma membrane. So just to summarize, the chemical gradient is going to be the concentration gradients of ions, specifically usually sodium and potassium, whereas the electrical gradient is going to be charges that are separated by the cell membrane, so whether it's more positive or more negative. And in typical format, the cytosol is usually much more negative than the extracellular fluid. And then when we combine the chemical and electrical gradients together, we have what's called the electrochemical gradient, which is the sum of both the chemical and the electrical forces acting together on an ion to allow passage across the membrane. And this is a form of potential energy. The equilibrium potential is the membrane potential at which there is no net movement of a particular ion across the cell membrane. So you'll see that for potassium, the equilibrium potential is negative 90 millivolts, whereas for sodium, the equilibrium potential is positive 66 millivolts. So earlier on, we just discussed the membrane potential of most neurons as being at negative 70 millivolts, which is much more similar to the equilibrium potential of potassium than it is from sodium, right? So for the equilibrium potential to meet the membrane potential of the neuron, we would have to add 70 plus another 66 millivolts, because that's the difference between the two. Whereas there's only 20 millivolts of difference between potassium and the membrane potential of the actual neuron itself. So the resting membrane's permeability to sodium is very low because the difference is so great, and so sodium has a very small effect on the resting potential. Now let's look at potassium and sodium ion gradients. So first, let's look at potassium. So what you'll see here in this image is a reminder that the resting membrane potential of the neuron is negative 70 millivolts, and recall that the equilibrium potential for potassium is negative 90 millivolts. And so when a neuron is at a resting membrane potential, the chemical and electrical gradients are different, right? So there's a difference of 20 millivolts between the two. So that difference, the net electrochemical gradient, tends to force the potassium ions out of the cell into the extracellular fluid. So we recall that plasma membranes are not just semi-permeable, they are selectively permeable. So they will allow only certain things in or out. But hypothetically, if a plasma membrane was freely permeable, specifically to potassium ions, then potassium would just continue to leave the cell until an equilibrium potential was reached, which is negative 90 millivolts. And keep in mind that's only 20 millivolts away from the actual plasma membrane potential. So although the equilibrium potential for sodium is very different than the resting membrane potential of the plasma membrane, There are forces more than just that at play. We have the net electrochemical gradient forces at play here. So collectively, the electrical charges and the chemical gradient is going to force sodium ions into the cell. So again, let's discuss the advantage of having a selectively permeable plasma membrane on our neurons. So if it was freely permeable, sodium would just rush into the cell because the sodium has an equilibrium potential of positive 66 millivolts, whereas the membrane is going to be at negative 70 millivolts. So that's a huge difference. So what that means is there would be a massive influx of sodium that would come into the cell, and that would clearly be pretty damaging for all of the organelles within the cell. So that plasma membrane plays a very important role of allowing certain ions to pass and in maintaining an electrical and chemical gradient. Now, one thing just to remember when you're trying to think of where is it positive and where is it more negative, there's a tip and tool in your textbook, and it shows you that you can associate negative with the inside. So the two ends, negative and inside, and positive on the outside. So positive has an O and outside has an O. So if you think of those together, that's a good little trick to keep in your mind. We previously discussed the passive processes across the plasma membrane of a neuron, but there are also active processes at play, and the sodium-potassium exchange pump is probably the most important one. So as we know, active processes are all powered by ATP produced by the mitochondria, and for every 3 sodium ions that are ejected, there will be 2 potassium ions brought in. And so what this does is it balances the passive forces of diffusion, which we recall are going to be different because of the different chemical and electrical gradients on either side of the plasma membrane. So the sodium-potassium exchange pump is just stabilizing that resting membrane potential to keep it at negative 70 millivolts. And this happens because the ratio of sodium entry to potassium loss through passive processes is 3:2. So the sodium-potassium exchange pump does the exact same thing in reverse to keep things stable. So why do we have this resting membrane potential? Well, it's there because there's a difference in the composition of the fluid, both ionically and chemically, in the cytosol as compared to the extracellular fluid, meaning there's a difference in the electrochemical gradients inside and outside the cell, and the plasma membrane is selectively permeable. So that membrane potential is going to change in response to various temporary changes in membrane permeability. So that means that there's going to be some opening and closing of various membrane channels, and it will do this in response to stimuli. So that membrane is constantly adjusting to be able to maintain an appropriate environment for the cell. The membrane potential is determined primarily by sodium and potassium. These are the 2 elements that are going to control the membrane potential. And they're going to be controlled by mechanisms that are either passive or active. So the passive ion channels are typically leak channels, and these are always open. And so the permeability will change with conditions, but they're always open, leaking small amounts of these ions. Active ion channels, such as gated ion channels, will open and close in direct response to various stimuli, but at resting membrane potential, most of them are going to be closed. So you can think of this as the leak channels being small streams that boats can pass on. So water is always constantly flowing and the boats can go by, but they're just narrow little small passageways, and they'll respond to if there's more water or less water. So if there's less water, then theoretically that stream might get too shallow for boats to pass. And then the active ion channels or the gated ion channels can be considered to be like the big locks that will allow things like a big ocean liner to be able to go through. So they'll hold that at bay and then open up the locks, and then the water and the big boat will come rushing through. But most of the time, those locks are going to be closed to stop anything from just passively going through. There are 3 general types of active channels that allow this passage. There are chemically gated ion channels, voltage-gated ion channels, and mechanically gated ion channels. We're going to discuss each of these next. Each of the 3 active channels are going to be exactly what you think of them as. So a chemically gated ion channel is going to open when it binds to a specific chemical. So for example, acetylcholine. So there's an image on the right, which is figure 1210 in your text that you can take a look at, that shows you how there's an ACH binding site up at the top, and a sodium ion comes along. It's not going to bind, but acetylcholine does. It binds, it opens the channel or closes the channel. So these are going to be found on the cell body and the dendrites of neurons, and they're quite common. Voltage-gated ion channels open and close and respond to differences in the membrane potential. So you're going to find these throughout the entire length of axons of neurons and also in the sarcolemma of skeletal and cardiac muscle cells. So the activation gate is going to open when it's stimulated, and it'll close to stop ion movement. And there are three possible states that can be happening at any given point with any one of these voltage-gated ion channels. It can either be closed but ready to open. It could be wide open, which is activated, or it could be closed and not capable of opening yet. Mechanically gated ion channels are only going to respond to membrane distortion, so something that physically actually pushes them to open. So you'll find these in various sensory receptors that are going to respond to things like touch, pressure, or vibration. A graded potential, or a local potential, is just a change in the membrane potential that can't spread very far from the site of which it started. So this is not an action potential which carries an electrical impulse from neuron to neuron. This is just changing the membrane potential locally in a very specific area. And this can be produced by anything that opens the gated channels. So once those gated channels open, there will be a change, and that will be a graded potential. So for example, if a resting membrane is exposed to some sort of chemical, then chemically gated sodium channels will open, then sodium ions will enter the cell, and the membrane potential will rise, which is depolarizing. Graded potentials might look tricky, but they're really not. So if you think about the sodium ions that are outside the plasma membrane, they're flowing along, and they're moving along parallel to it, and this produces a local current. So they're flowing along, and all of these sodium ions approaching the plasma membrane are going to depolarize nearby regions of the plasma membrane, creating a graded potential. So the change in this graded potential is going to be directly proportional to the stimulus. So a stronger stimulus would equal a larger graded potential versus a smaller stimulus will have a more localized and even smaller graded or local potential. So in a graded potential, any shift away from the resting membrane to be less negative is going to be a depolarization. Whereas when the stimulus is removed, then that membrane potential is going to return back to normal. So we'll say that it became repolarized through a process called repolarization. Hyperpolarization is going to result from opening those potassium ion channels so that all the potassium ions move out, but they don't move into the cell. So this has the opposite effect of opening the sodium ion channels, which would increase the positivity of the resting potential, but the potassium ions are going to increase the negativity of the resting potential. I'll explain this more with an image that'll show you what I mean in just a moment. Figure 12-12 in your text shows you how depolarization, repolarization, and hyperpolarization look on an electrical level. So the first thing I want to point out to you is that when you're looking at this graph, you'll see that the y-axis shows you time and the x is showing you the membrane potential in millivolts. But please note that it's all in negative, right? So if you look up at the higher points of the chart, we actually have a lower millivolt number. And as you go down, we become more negative. So just keep in mind that we are negative here, not talking in terms of positive integers. So at the very beginning of our baseline, we're at negative 70 millivolts, which is our membrane potential, and a chemical stimulus is applied, which results in the cell depolarizing because it moves closer towards a less negative potential. It moves towards 60 as opposed towards 80. So it's moving towards 60, it's becoming less negative. So then when the chemical stimulus is removed, the membrane potential is going to return back to normal, which is a more negative baseline at negative 70 millivolts, and we call that repolarization. So now we're at our resting membrane potential again, but a different chemical stimulus is applied over on the right. And you'll see here that the line dips down closer towards 80, so it's actually becoming more negative, and that's considered hyperpolarization. And then once we finally remove that stimulus, then it will return to a resting membrane potential and be repolarized. So some of the characteristics of graded potentials are that the membrane potential is going to be changed most at the site where the stimulation occurs. And then the further away you get from the site of the stimulation, the effect is going to decrease. So it's like if you're walking away from something hot, it gets cooler and cooler as you move away. This is the same sort of idea here. So the membrane potential will be strongest at the site of stimulation and then weaker further away from it. So the effect of this membrane potential is going to spread very passively just through local currents passing ions along the exterior surface of the cell or along the interior surface of the cell. The graded change in membrane potential can involve depolarization or hyperpolarization, with the idea being that to return to normal, it would repolarize. And a stronger stimulus will produce a greater change, and so therefore it will affect a larger area, whereas a smaller stimuli is going to produce a smaller change in the membrane potential and affect a smaller area. Graded potentials are often going to trigger specific cell functions. So for example, exocytosis or glandular secretions are contingent upon graded potentials to allow them to expel their product. Acetylcholine actually causes the graded potential at the motor end plate of the neuromuscular junction, so this has an important role in movement. Pop quiz time. Voltage-gated channels open in response to A, a change in the electrical potential, B, neurotransmitter attachment, C, sodium diffusion, or D, potassium diffusion. If you thought a change in the electrical potential, you're right. That one might have been a bit too easy. Let's try a few more that are a little trickier. Chemically gated or ligand-gated channels open in response to A, a change in the electrical potential, B, neurotransmitter attachment, C, sodium diffusion, or D, potassium diffusion. Well, this one might be a little trickier, but I'm going to guess that you probably got it. So we remember that there are three different types of channels that would open. One is going to be chemically gated or ligand gated. The other is voltage gated, which we just discussed in the previous question. And the third is mechanically gated. So mechanically gated are going to open in response to physical distortion of the membrane surface, whereas the voltage gated ion channels are going to open or close in response to changes in the membrane potential, right? Because it's a voltage gated. And then we have the chemically gated ion channels or ligand gated ion channels. And these are going to open or close when they run into specific chemicals that they bind with. And these are usually ligands. And specifically, you'll see acetylcholine being one of the most common. So in this case, the answer is B, neurotransmitter attachment, because that qualifies as a ligand that would bind to allow the channel to open. The concentration gradient for sodium in a resting neuron is A, from the extracellular fluid to the intracellular fluid, B, from the intracellular fluid to the extracellular fluid, or C, there is no concentration gradient for sodium. Well, we know that we can rule out C for certain because we absolutely have a concentration gradient, and this is an important part of action potentials being able to be generated. So if we think back to how an action potential is generated, we know that when the sodium ion channels open, we have a rapid depolarization as the sodium rushes into the cell. So when we think about that, that means if the sodium is rushing in, it's seeking to eliminate that concentration gradient, right? So we know that that means there is more sodium on the outside of the cell than there is on the inside of the cell. So the answer is A, from the extracellular fluid to the intracellular fluid. Now let's look at the inverse type of question. The concentration gradient for potassium in a resting neuron is A, from the extracellular fluid to the intracellular fluid, B, from the intracellular fluid to the extracellular fluid, or C, there is no concentration gradient for potassium. Well, since we know that sodium and potassium have opposite relationships in regards to the cell and to action potentials, you could probably guess right away that's the exact opposite of the previous question, and you'd be right. But it's important to note that in an action potential, we have that brief moment of hyperpolarization, and that is at the very end of the action potential, and it's because we have those voltage-gated potassium channels that start closing, but as they're closing, there are still potassium ions leaking out. And so as that happens, we have our brief hyperpolarization. So the answer is B, from the intracellular fluid to the extracellular fluid. Voltage-gated ion channels are found on the blank of a neuron. A, cell body, B, dendrites, C, Schwann cell, or D, axon. So let's close our eyes for a minute and imagine your typical multipolar neuron. In a multipolar neuron, you're going to have a large cell body with multiple dendrites extending from it and a long single axon extending off one end with multiple Schwann cells wrapped along the axon if it's an unmyelinated axon. So where would those voltage-gated ion channels be? Well, let's think about what happens with an action potential. So the graded potential arrives, and it is going to affect the axon because that's what transmits the action potential, and also parts of the axon hillock and initial segment. So when you look at the options here, you have to figure which is the best answer. And in this case, the correct answer is D, axon. Let's discuss action potentials now. It's section 12-5 in your text. All right, we finally get to talk about action potentials. These are also known as nerve impulses, and it's passing energy from neuron to neuron. And it's propagated by changes that occur in the membrane potential. So all of that we just talked about will lead us up to the possibility of an action potential actually occurring. So in an action potential, the entire excitable membrane will be involved, and it begins at the initial segment of the axon, which makes sense, right? It begins at the beginning, the initial segment, and it doesn't diminish as it moves away from the source. So what I'm saying here is that the action potential, once it generates that energy, it continues to keep propagating from cell to cell. So what this means is this is different from the graded potential, which you can consider to be more like the way a wave on a regular ocean would rise and crest, and the further it goes, the more it just dissipates. An action potential is more like a tsunami. Once it gets moving, it just continues moving and it maintains its strength from cell to cell. So this is stimulated by a graded potential that's going to depolarize the axolemma, which is the outer aspect of the axon, and it's brought to threshold because the action potential is an all or none activity. It's either going to fire or it's not, and that's it. So the threshold for an axon is -60 to -55 millivolts, and keep in mind the standard expected baseline would be -70 millivolts. So we have to have a difference of at least 10 to 15 millivolts to actually hit threshold so that an action potential can occur. The first thing to know about an action potential is the all or none principle. So basically all this means is either you have an action potential or you don't. There's no such thing as 50% of an action potential. It's like you're either pregnant or you're not, right? So with the all or none principle, what this really means is any stimulus that's going to change the membrane potential all the way to threshold will trigger an action potential. If we are even just a tiny bit off from trying to get to threshold, it will not cause an action potential. And if it's way over the threshold, it still causes the same action potential. So all action potentials are going to react in the same way. So there's no such thing as a big action potential or a small action potential. We can only have more of them more frequently. So it doesn't matter how large the stimulus is, they're all going to be the same. And an action potential, as its final take-home point here, is either triggered or it's not. So there are a couple examples in the textbook. One is about flushing the toilet. Another one is pulling the trigger on a gun. But the concept is the same. You can pull on the trigger of a gun and pull, nothing happens. But when it finally reaches the threshold of pressure, then the gun triggers and the bullet is ejected. Or with a toilet analogy, if you're flushing the toilet, you're pressing down on the lever and nothing happens up until the point where finally it's triggered. And once it starts, you cannot stop it. It's going to flush the toilet. So that's the whole concept of the all or none principle. Every neuron is going to have graded potentials all over the cell body, the dendrites, everywhere. And if those accumulate in a strong enough trigger to actually reach threshold, then we will trigger an action potential. So the action potential is going to be a propagated change that once it starts, it doesn't stop. So it's going to collectively gather all of the information at the axon hillock. And if an action potential is fired, it starts in the initial segment, as you can see in figure 12-13 above. So now let's walk through the steps of what happens once an action potential actually starts. Let's discuss how an action potential is generated. So the first thing I want to discuss with you is that although we previously talked about chemically gated channels and mechanically gated channels, the only channel that's really relevant with an action potential is the voltage gated sodium and potassium ion channels. So let's start off with our resting membrane potential. We're at -70 millivolts, and at this point we are ready to receive depolarizing stimuli. So there will be a number of stimuli that arrive at the cell, and this will basically create a graded depolarization. And if it doesn't reach the threshold, then nothing happens. And remember, graded depolarizations are just basically like a wave that's going to dissipate with time and distance. But on the other hand, if we have a stimulus that initiates a graded depolarization that's large enough to actually trigger the threshold point, then we will have an action potential begin. So let's say that we actually meet our triggered threshold and an action potential is about to occur. At this point, we're going to have the sodium channel activation gates open up, and then the plasma membrane will have a lot more sodium rush into it. So because of this large electrochemical gradient, the sodium ions come into the cell, and this depolarizes the cell very quickly. So when this happens, the inner membrane is going to have more positive ions, and then the membrane potential is going to change from negative 60 millivolts to a positive value. So this is massive because when those gates open, in comes the sodium. So now what ends up happening is we have to close these gates. So we inactivate the sodium ion channels, and the activation of potassium ion channels starts to open up, and this starts repolarization. Now we also have the hyperpolarization that we see as the end point of all action potentials, and this is because there's a lag time in when the potassium ion channels close. So for a little while we still have some potassium moving, but it's once they close that we'll return back to our resting membrane potential. The refractory period is just a point at which the membrane won't respond normally to any additional stimuli. So this is the time between when an action potential starts to when it returns back to its resting state. So it's just not responding normally at that point. There are two subdivisions of the refractory period. There's the absolute refractory period and the relative refractory period. The absolute refractory period means that membrane can't respond, period. It's absolutely not going to respond to any stimulus no matter how strong it is, because all of the voltage gated sodium channels are already open or they're inactivated. So there's nothing further that can happen. So that's it. Now the relative refractory period is meaning that a strong stimulus can actually initiate another action potential. So this begins when the sodium channels start regaining their resting condition, but it's not fully resting yet. But because it's starting to rest, what that means is that these gated channels are starting to close, and so that means that a strong stimulus can actually open them again and allow another action potential to occur. So the image that you see on the right is figure 1213 in your text, which shows you how this works. So let's walk from left to right. So we start off at left with our resting membrane potential at negative 70 millivolts. And then we have a graded potential, which brings the cell to threshold. So once we hit the threshold, an action potential is triggered. So now we'll move into depolarization, and all of the voltage-gated sodium channels will open up, and the sodium ions are going to move into the cell really rapidly. So we move from a depolarized negative 70 up to a positive 30 millivolts at this point. So we're way above zero. And then further, we have our absolute refractory periods and relative refractory periods that you can see at the very top of this schematic. And so as you can see in red, the absolute refractory period is at the beginning of the process in which nothing further can happen. The gates are already open. There's no more gates to open. There's nothing more to do. So even if additional stimulation comes along to that cell, the neuron cannot respond any further. But then as the sodium channels begin to close, then we have a possibility for the relative refractory period. So theoretically, as that is dropping, if there was a strong stimulus that came along, we could theoretically have another action potential triggered. Propagation is how a neuron moves an action potential along the axon in a series of steps so that it can continue that action potential to keep going. There are two different types of propagation, continuous and saltatory. We're going to discuss those in the next couple slides. In continuous propagation of action potentials, the action potential is repeated over and over and over again, just in the same format. And it can't move backwards because the cell will be in the refractory period, specifically the absolute refractory period, so it can't open up those gates. Once they're closed, the action potential has no choice but to only move in one direction. So let's use the analogy of the passing the message on in the movie line that you'll see in your textbook. The idea of a continuous propagation is that one person would tell the person behind them, the movie starts in 5 minutes. Pass it on. So then the second person tells the third person tells the fourth person tells the fifth, and on it goes, right? So it'll just keep moving along, and this is a process that we'll see in unmyelinated axons, and it actually goes as quickly as up to 1 meter per second, which is pretty darn quick. So now let's look at the next type of propagation. In saltatory propagation of action potentials, we have a faster process. So this occurs in myelinated axons to begin with, which we remember are insulated and also just faster conductors in the first place. And this actually requires less energy than continuous propagation. And here's why. We're going to use that same model of passing along information in the movie theater, but instead of person 1 telling person 2 and person 2 telling person 3, there might be, let's say, 5 or 10 feet between them. So that information doesn't have to go just the 1 or 2 feet from person to person. These could be socially distanced individuals quite far away from one another. So the distance that's covered is actually much faster because each message will go further. So the myelin is the substance that's going to prevent it from being continuously propagated. So the local current has to jump from node to node. And this depolarization, by the way, only occurs at nodes. By the way, one of the things I always think about when I'm studying this is saltatory is a term that comes to mind pretty quickly for me. My hometown is called Sault Ste. Marie, which is a French name. I'm from Canada originally. And Sault is French for sault or jump. So basically, saltatory is jumping propagation. So to me, that was always a quick thing to remember. That might not be as easy for you, but nonetheless, I thought I would share with you the way that I learned to remember this. The diameter of the axon is going to be what affects the propagation speed of action potentials. So the big rule of thumb here is the larger the diameter, the lower the resistance, and the faster the speed. So if you want to conduct a really strong electrical current, you would use a big wire, not a little tiny wire, right? That's the general idea here. So we're going to break down different types of axons based on their diameter, their myelination status, and their propagation speed. Type A fibers are your fastest fibers, and these are myelinated and large diameter. And because myelin helps to facilitate speed, and the large diameter also does too, these are able to transmit information really quickly to and from the central nervous system. So it transmits information that has to be transmitted very quickly because it's really important to our life and death. So it will include sensory information such as our position and balance so that we don't fall off a cliff, and also motor impulses to our skeletal muscles so that we can move quickly and prevent injury. Type B fibers are myelinated also, but they're a smaller diameter than the type A fibers. So these are going to transmit information at more intermediate speeds, which is roughly about 18 meters per second. Type C fibers are our slowest. They're unmyelinated and they are small diameter, so they have both things going against them in terms of speed. So they're going to transmit information quite slowly at about the rate of 1 meter per second. So most sensory information is going to be transmitted by a type C fiber. So think about when you're eating dinner and you're tasting your food, that's not necessarily an emergency. So you can have type C fibers that are going to help us to sense things like that. But things that are immediate to our life and death are going to be type A fibers. So the way I think about this is in terms of a personality test. So we know our type A personalities are those people who are very ambitious and driven and they're focused, and they are basically like the big brother or the big sister of the family. So the type A fibers are the ones that get it done and they get it done quickly. And the type C fibers, they're kind of laid back. They're like the baby of the family. They're going to get the information there, but they're going to do it on their own time. And the type B fibers are kind of like the well-adjusted middle child, right? They're going to have that medium diameter size, and they're also myelinated. So they have some things going for them. They're fully capable of doing things, but they're doing it at an intermediate speed in between the type A and the type C fibers. So I always think about this as like a family tree, and I hope that helps you to remember this too. Now let's talk about synapses. This is section 12-6 in your text. Synapses are the space between a neuron communicating with another cell. So they have to send the information via action potential from one neuron to another cell. So the one that is sending the message is going to be called the presynaptic neuron. And then if there's a second neuron that's receiving the message, that will be called the postsynaptic neuron. There are two types of synapses, electrical and chemical. Electrical synapses are actually quite rare, and they require direct physical contact between the cells. Now, chemical synapses are much more common. In this case, what we have are two cells that are very close, but the signal is going to be transmitted across the gap, across the synapse, by neurotransmitters. So this is responsible for transmitting a ton of information. Electrical synapses, while rare, do exist, and so the way that they're held together is that the presynaptic and postsynaptic membranes are locked together at the gap junctions, and so the space between them may only be 2 nanometers, so this is quite small. Ions are going to be able to pass between the cells and through the pores, and there are integral membrane proteins that are going to bind these positions in place. Those integral membrane proteins, by the way, are called connexons. So there's going to be local currents on both cells that are going to affect them both, and action potentials will be propagated quite quickly with electrical synapses. You can expect to find electrical synapses in some areas of the brain, but mostly the eye and in the ciliary ganglia. The chemical synapses are the most common type of synapse. So this is the most common type of space between neurons. And it is the only type of synapse that allows for a neuron to communicate with a different type of cell and not just a neuron to another neuron. So the space between them is called the synaptic cleft, and the presynaptic cell is going to be the one that is sending the message, and the postsynaptic cell is going to be the one receiving the message. Figure 12-15 in your text shows you what a typical chemical synapse looks like. So starting down at the bottom, that broad gray area is going to be our distal cell. So this is the one that's going to be receiving the message and the action potential. So at the bottom, we have the postsynaptic membrane that is covering the postsynaptic neuron. The space in between them is the synaptic cleft. And then that broad pink structure is going to be our axon terminal. And at the very end of the axon terminal, you can see that there are little green balls inside of that. These are synaptic vesicles, which will be secreting neurotransmitters. And so the little structure that you see that is the purple that encircles all the way around that cell is the presynaptic membrane. Moving up a little bit further, you can see that there's a mitochondrion. I think that's quite clear. And then we have the axon terminal, and at the very, very top, the teledendrion. In addition to chemical synapses that are in between two different neurons, there are also chemical synapses that will communicate between two different types of cells. So the neuromuscular junction chemical synapse is going to be the synapse that is in between a neuron and a skeletal muscle cell. So in this case, the neuron is going to be the presynaptic cell, and the skeletal muscle cell will be the postsynaptic skeletal muscle cell, because the synapses between them, the neuron comes first, and then the skeletal muscle cell comes afterwards. There's also the neuroglandular junction, and this is a type of chemical synapse in which the synapse is in between a neuron and a glandular cell. So in this case, the neuron will be the presynaptic neuron, or the presynaptic cell, and then the gland cell is going to be the postsynaptic cell. Neurotransmitters are chemical messengers that are held in these synaptic vesicles in the axon terminal of the presynaptic cell. Another way to think of this is at the very, very end of the cell that is sending the neurotransmitter, they are inside the end in little packaged bundles called synaptic vesicles, and they are ready to burst out and release into the synaptic cleft, or the space in between the presynaptic neuron and the postsynaptic neuron or postsynaptic cell. So neurotransmitters are going to affect the receptors of the postsynaptic membrane, which will then be broken down by enzymes. Also, inside the presynaptic cell, any breakdown products that are present as a part of having the neurotransmitters inside the axon terminal will be reabsorbed and reassembled. Also, that distal part of the axon terminal, is going to be receiving a variety of different substances that are brought in by anterograde flow. And so some of those substances will be things like enzymes and lysosomes to be able to break down and reassemble any byproducts that are floating around in that space. Neurotransmitters that are from the presynaptic neuron are going to bind to the postsynaptic plasma membrane. And when this happens, it's going to produce a localized change in both the permeability and the graded potential of that postsynaptic neuron's plasma membrane, or the postsynaptic cell's plasma membrane. So there may or may not be another action potential that is generated in the postsynaptic cell. And this will depend on if there is enough neurotransmitter released, and also the sensitivity of the postsynaptic cell. So any arriving action potential will definitely arrive at the postsynaptic cell, but that is no guarantee that the postsynaptic cell will also, in effect, have an action potential. It will be dependent upon the receiving cell's ability to process the information it received and whether or not it rises to a threshold to create another action potential. Cholinergic synapses are a great example of a chemical synaptic function because they release acetylcholine, which is the very most common and widespread neurotransmitter. We abbreviate this ACH. So it's found in all sorts of places, ranging from all of the neuromuscular junctions, which includes skeletal muscle fibers. It's also present in many synapses in the central nervous system. It's present in all of the neuron-to-neuron synapses in the peripheral nervous system, and also it's present in all neuromuscular and neuroglandular functions in the parasympathetic division of the autonomic nervous system. Figure 12-16 in your text shows you the events that are happening in a cholinergic synapse. We're just going to go through this very briefly before we look at it in a little more detail with the images. But just basically, an action potential is going to arrive at the presynaptic neuron, and as that action potential arrives, it's going to depolarize the membrane. And depolarizing the membrane is going to open up calcium channels, so that way it's going to trigger exocytosis of acetylcholine from the presynaptic neuron. The acetylcholine is going to diffuse across the synaptic cleft, and it binds to the receiving cell and depolarizes it. And if it creates enough of a depolarization, then it can create an action potential, but that's not a guarantee. And then the acetylcholine that's remaining is going to be removed from the synaptic cleft by acetylcholinesterase, or abbreviated ACHE. And just by the way, ACHE breaks acetylcholine into acetate and choline. Figure 12-16 in your text shows you 4 different images that show you the process of a cholinergic synapse. So in the very first image, an arriving action potential is going to depolarize the axon terminal. This is going to set the stage for a number of different activities. As the action potential arises, you'll see that the membrane changes color on the presynaptic cell. So when this happens, you're going to see voltage-gated calcium ion channels open, and because of that, calcium ions are going to rush into the cytosol of the axon terminal. All of this calcium coming in essentially makes the cell throw up. So it basically sends out all of its acetylcholine in all of those little synaptic vesicles by exocytosis. So you can see those little vesicles at the very bottom of the presynaptic membrane, and they're labeled ACH. Once the acetylcholine has been spit out from the presynaptic cell, it's going to diffuse across the space between the two cells, which is called the synaptic cleft, and it's going to bind to receptors on the postsynaptic membrane. Because of this, cation channels are going to open, specifically for sodium and potassium, and this helps produce a graded depolarization. Finally, the depolarization is going to end because the acetylcholine is going to be broken down into acetate and choline by acetylcholinesterase. The axon terminal is also going to reabsorb choline from the synaptic cell cleft, which is the space between the two, and it's going to use it to resynthesize more acetylcholine. There's a slight delay in between the time that an action potential arrives at an axon terminal and when the neurotransmitter actually affects the postsynaptic cell. We call this the synaptic delay. Now, at first blush, you would probably think that the reason for this is because it takes time for the neurotransmitter to diffuse from the first cell to the second cell. But that's not actually the reason why. The reason why is it takes time for the calcium ions to flow into the axon terminal of the presynaptic neuron, and then for that presynaptic neuron to realize that it's time to spit out the acetylcholine. Another interesting point about synaptic delay is that if we have fewer synapses, we'll have faster responses. So, for example, things like reflexes, which are really important for making sure that we stay alive and don't get injured, there may only be one synapse, and this allows us to be able to move very quickly and to respond to things that could injure us. There's also a concept called synaptic fatigue, which is essentially when a synapse just can't even. It's just had it. So what's happening here is the neurotransmitter just can't be recycled fast enough to meet the demands of any more stimuli. So it's just going to weaken the response of the synapse until acetylcholine is replenished and it can return to normal function. All right, let's talk about neurotransmitters and neuromodulators. This is section 12-7 in your text. Neurotransmitters are going to be substances that are transmitted from one nerve to the next. Neuromodulators are substances that will change the way that the cell responds to the neurotransmitter. So let's talk about the different types of neurotransmitters first. So we have excitatory neurotransmitters, and these are going to cause a depolarization of the postsynaptic membranes. And so because it's depolarizing, that's helping to create a graded potential which will promote action potentials. We also have inhibitory neurotransmitters, and these are going to do the exact opposite. Instead of causing depolarization, they'll cause hyperpolarization of the postsynaptic membranes, and this actually acts to suppress action potentials, meaning that it will take even more of a stimuli for the cell to have an action potential. One important point about the effect of neurotransmitters on postsynaptic membranes is that the effect is completely contingent upon the properties of the receptor. It's not on the nature of the neurotransmitter. You'd be inclined to think that if a certain neurotransmitter is released, that there would be a standard response, but that effect can be varied contingent upon the properties of the receptor on the receiving cell or the postsynaptic membrane. There are major classes of neurotransmitters which we'll discuss briefly, such as biogenic amines, amino acids, neuropeptides, and also dissolved gases can act as a neurotransmitter. Here are some biogenic amines that you're probably already familiar with. Norepinephrine is also known as noradrenaline, and is released by adrenergic synapses. It can have an excitatory and a depolarizing effect, and is widely distributed throughout the brain and portions of the autonomic nervous system. Dopamine is a central nervous system neurotransmitter, and it can be excitatory or inhibitory, and it's involved in Parkinson's disease and also has a major role in cocaine use. There's current research being done as we speak that involves the effect of long extended use of cocaine and the effect on dopamine stores. And the idea is basically that dopamine stores may be depleted permanently to some extent after somebody has had extensive cocaine use. Serotonin is another biogenic amine that you're probably familiar with already. And there are a lot of antidepressant drugs on the market, and many of them are SSRIs, or selective serotonin reuptake inhibitors. And what they're doing is basically just trying to increase the serotonin concentration at the synapses because it's preventing it from being reabsorbed. Gamma-aminobutyric acid, abbreviated GABA, has some implications in being able to reduce anxiety, but it's still being studied. It also has an inhibitory effect as well. Neuromodulators are chemicals that are released by the axon terminals on the presynaptic neurons, and these chemicals are going to alter both the rate of the neurotransmitter release or potentially the response by the postsynaptic cell. The effects of neuromodulators take some time to appear, and they're long-term. And so the responses to this are going to include multiple steps and intermediary compounds, and we're not going to get into the weeds on that. But the neuromodulators will affect the presynaptic membrane or the postsynaptic membrane, or even both. It's also worth noting, by the way, that neuromodulators do not operate in a vacuum. They could be released alone, or they also could be released with a neurotransmitter as well. Neuropeptides are one type of neuromodulator, and these are really just small peptide chains that are synthesized and released by the axon terminal. Opioids are one such type of neuropeptides, and these are obviously very important in pain control for people. So oid meaning resembling, and opi being opiate. So we're talking about opium and morphine specifically. So an opioid is a drug that is similar to opium or morphine, and it's going to bind to the exact same receptors as opium and morphine. So several classes of opioids that are present in the central nervous system are enkephalins, endorphins, and dynorphins. It's worth noting that there are dissolved gases that actually function as neurotransmitters. Nitric oxide is one such dissolved gas, and it's important in that it innervates smooth muscle in the walls of blood vessels. So what this means is it can be involved in vasoconstriction and vasodilation. Carbon monoxide is another important neurotransmitter that is a dissolved gas. Neurotransmitters and neuromodulators may affect the distal cell in a number of ways. So here are three of them. The first is a direct effect. The second two are both indirect. So the first one is a direct effect on the membrane potential of the postsynaptic cell. And this is by opening or closing the chemically gated ion channels. Examples of this include acetylcholine, glutamate, and aspartate. The two indirect options are basically going to work through either G proteins or through intracellular enzymes. So G proteins will be helpful in working with norepinephrine, dopamine, serotonin, histamine, and GABA. The other indirect effect is going to work through intracellular enzymes. So the lipid-soluble gases here, like nitric oxide and carbon monoxide, are included. Lastly, let's look at neurons and how they integrate both excitatory and inhibitory stimuli. Let's discuss information processing of a receiving neuron. So any individual neuron is not just receiving a single chemical. They'll be receiving a bath of multiple different chemicals mixed in. So when this happens, that receiving cell, or the postsynaptic cell, is going to have to integrate all of this stimulus and figure out what to do with it. So at the simplest level, we can look at just individual neurons. So let's look at just one individual neuron. It's going to have a whole bunch of dendrites, right, on the receiving end, so it's able to receive neurotransmitters from multiple different places all at the same time. And some of those are going to be excitatory and some of them will be inhibitory. But the net effect is going to come together at the axon hillock just before the initial segment of the axon. And if there is enough of a graded potential that occurs as a result of all of these different chemical interactions, if it rises to the threshold to generate an action potential, then that will determine whether or not an action potential is actually produced. Postsynaptic potentials are really just graded potentials that have developed in the postsynaptic cell. So it's received a whole bunch of different neurotransmitters, and as a result, we have a graded potential, which we'll just call a postsynaptic potential. And there are two different types of postsynaptic potentials: excitatory postsynaptic potentials or inhibitory postsynaptic potentials. And so in an excitatory postsynaptic potential, we are increasing the chance of another action potential to occur. So that means we are further depolarizing. So this is a graded depolarization on the postsynaptic membrane. The inhibitory postsynaptic potential does exactly what you think it does. It inhibits the chances of a potential action potential. And so what we're having here is, instead of a depolarization, which would make it easier for an action potential to reach threshold, we have a graded hyperpolarization, which does the exact opposite and makes it much less likely and much more difficult for that cell to actually rise to the threshold to trigger an action potential. Let's imagine a single neuron for a moment. So this neuron is a receiving neuron that is receiving the bath of chemicals coming from the presynaptic cell. This is going to include neurotransmitters, neuromodulators. And so let's say that overall the majority are IPSPs, or inhibitory postsynaptic potentials. So the inhibitory postsynaptic potentials are going to inhibit that cell from producing an action potential because of hyperpolarization. So because it's hyperpolarized, it's going to make it even more difficult for threshold to be reached. So that way that when the threshold is reached, we can trigger an action potential. So this moves us further away from the threshold or the trigger. So in this case, to trigger an action potential, one excitatory is not going to be enough. We're going to need multiple, and we can do it in a number of ways. So we do this through a process called summation, in which we're going to add up our excitatory postsynaptic potentials and also the inhibitory postsynaptic potentials as well. But ultimately, we're going to come together to create another action potential. So one way is through temporal summation. And temporal summation is just over time. So basically, it's repeatedly over and over and over and over. Effectively, temporal summation is nagging over time, if you want to think of it that way. And then we have spatial summation, which is when you have multiple different stimuli all arriving at multiple synapses, and maybe each individual one isn't enough to make anything happen, but all together, the force of their numbers will basically force the potential for an action potential to occur. Figure 1218 in your text shows you temporal summation and spatial summation. In general, I think of temporal summation as the nag approach, where over time, basically you ask for an action potential over and over and over, and eventually get your way. Now with spatial summation, we're looking at the power and numbers approach, where multiple stimuli together are going to force an action potential. So here's how it works. In temporal summation, the first stimulus is going to arrive, but it's not enough of a depolarizing event to create an action potential. But when the second stimulus comes along, it's going to build the amount of depolarization that's present on the membrane of the cell, and therefore we can reach the threshold to trigger an action potential so it can be propagated. In spatial summation, we have two or more stimuli arriving at the same time, and so because of this, each of them on their own may not be strong enough to create an action potential, but together the depolarization will overlap, and eventually the threshold will be reached, and an action potential can be propagated that way. So as multiple excitatory postsynaptic potentials arrive and accumulate, the neuron is going to get closer and closer to actually reaching threshold and triggering an action potential. So the neuron in this case is considered facilitated because it's getting closer to actually having an action potential. And so finally, the excitatory and inhibitory postsynaptic potentials that are neuromodulators and hormones can change the membrane sensitivity to the neurotransmitters as well. So this shifts the balance between the excitatory and inhibitory postsynaptic potentials. Up until this point, we've discussed the relationship between presynaptic neurons, the synapse between them, and the postsynaptic neuron as if it's a perfectly linear A to B to C type relationship. But we know from studying earlier that there are a lot of different types of neurons, and they have different structures, including multiple axons sometimes, or multiple dendrites. So we know that there's got to be more than just an A to B to C linear relationship among neurons. So if you look at the image here on the right, it's figure 12-20, you'll see at the very top there are two neurons. One is green and one is yellow. So both of those are going to come together having their axons meet. So they have a relationship first before they interact with the postsynaptic membrane or the postsynaptic neuron, which is the purple cell that we see below. So we call this an axoaxonic synapse. And when those two axons come together, they can affect what's going to happen with the postsynaptic membrane. So there's either an inhibitory effect or a facilitating effect. So in the facilitating effect, all that's happening here is that the activity at the axoaxonic synapse is going to amp up the amount of neurotransmitter that's released when the action potential arrives. And the exact opposite is true with the presynaptic inhibition. So when that action potential arrives, it's going to decrease the amount of the neurotransmitter that's released at the presynaptic membrane. In some instances, the way that information is processed is just by the frequency or the rate of action potentials received. So this can be modified, of course, in a number of different ways, and this will depend upon the degree of depolarization above threshold. So if we have a cell, a neuron specifically, with a high level of depolarization that remains above threshold, it's basically the same thing as having a second large stimulus come along. So it keeps triggering action potentials for as long as it's held above threshold. Now that said, there is technically a theoretic maximum rate of action potentials, and that's reached when the relative refractory period is eliminated. But it's definitely worth noting that if there is just, let's say, one action potential coming in every second or so, that might result in just a little twitch or maybe a twinge of something that you feel in your muscles. But if they're coming in at a rate of, let's say, 100 per second or more, it will hold that muscle in sustained contraction called tetany. So the rate of these action potentials has a direct effect on the outcome of the tissues that are responding to it. So these are the big take-home points of the chapter. So we know that information is relayed in the form of action potentials, and this is with a graded depolarization that will then trigger an action potential if it rises to threshold. If it doesn't, then it's just like a wave that will dissipate over time and be ready for the next incoming stimulus. Neurotransmitters are released at a synapse, and these can have excitatory or inhibitory effects on the postsynaptic tissue. There are also neuromodulators, and these affect the way in which neurotransmitters will release or respond to the postsynaptic neuron. And neurons can either be facilitated or inhibited by chemicals that are other than neurotransmitters or neuromodulators. Remember, we talked before about some gases and other substances. And lastly, we note that the postsynaptic neuron, so this is the one receiving the message, the action potential, it can be affected by neuromodulators or a number of other chemicals that can cause both facilitation or inhibition. It can also be affected by activity that's underway at other synapses. So remember, we just discussed axoaxonic synapses. And it can also be affected by modification of the rate of the neurotransmitter release through either facilitation or inhibition. Okay, another pop quiz. If two EPSPs occurred simultaneously from different channels, what type of summation has occurred? Is it A, temporal, or B, spatial? Well, I always go back to my idea of thinking that temporal is like nagging, so it's like asking over and over and over again the same question. Whereas spatial allows for power and numbers, right, to occur at the same time. So the key word here is simultaneously in this question. So if two are occurring simultaneously, they can't be happening in tandem one after another. It's happening at the same time. So that means the correct answer is B, spatial summation. If a second identical EPSP occurs immediately after the first, will an action potential be generated? A, yes, or B, no? And then follow-up question, what type of summation is this? Is it A, temporal, or B, spatial? Well, before we go into the first question, I think we can answer the second one right away. We just discussed temporal and spatial summation, and we know that temporal occurs over time and spatial occurs with multiple occurring at the same time over different space. So we know that this is a temporal summation. But let's go back up to the first question. So if two EPSPs occur one after the other, will there be an action potential? And the options here are yes or no. And really what we need is an option of C, maybe, because truly we can't know because I didn't give you enough information in this question to answer the question fully. And the reason for this is if the EPSP is a small graded potential and the second one is another small graded potential, it's possible that those two graded potentials together will not rise to the threshold to trigger an action potential. But that said, it's altogether possible that the two EPSPs have enough of a graded potential that cumulatively they can rise to the threshold and actually trigger an action potential. So in this case, I would like an answer of C, maybe, but it's not here right now. But this is just the sort of thing I want you to think about before you approach the exam that covers this chapter. Okay, here's a much more specific question. So one EPSP depolarizes the initial segment from a resting potential of negative 70 millivolts to negative 60 millivolts, and the threshold is negative 55 millivolts. Will an action potential be generated? A, yes. B, no. Well, we know that negative 70 millivolts is a standard resting potential for a neuron, and the threshold is negative 55 millivolts, so we know that we need to have a difference of 15 millivolts in order to trigger an action potential to reach the threshold. In this event, the EPSP only depolarized the initial segment 10 millivolts and didn't bring it all the way to 15 millivolts to trigger that action potential. So in this case, the answer is no, an action potential will not be generated. This question is going to require you to do a little bit of math. So hot tip, you can expect to see stuff like this on the exam. So will there be an action potential generated? And the given situation is that the resting potential is negative 70 millivolts, the threshold potential is negative 55 millivolts, and what the cell receives are 3 EPSPs of 10 millivolts and 1 IPSP of 5 millivolts. So this is pretty easy math, but let's break it down. So we know that those 3 EPSPs of 10 millivolts, if we multiply 3 by 10, we have 30 millivolts, right? And so that's going to be 30 depolarizing millivolts. And 1 IPSP of 5 millivolts is going to be 5 millivolts of hyperpolarization. So let's put those together. So 30 and the 5. So we're going to subtract the 5 from the 30. So we have a difference here of 25 millivolts that are going towards the depolarizing end of things. So we know that we have to depolarize from 70 millivolts at the resting potential to get to 55 millivolts in order to trigger an action potential. So in this case, we're actually going to subtract from 70 our 25 and come up with negative 45 millivolts. So we not only met the threshold potential of 55 millivolts, but we were actually even over by another 10 millivolts. So yes, there will absolutely be an action potential generated. Here's another similar example. You receive 6 EPSPs of 5 millivolts each and 1 IPSP of 5 millivolts at the same time. Will you generate an action potential? And then the criteria here for this question is that the resting potential is negative 70 millivolts, and in this question, the threshold potential is negative 50 millivolts. So will there be an action potential generated? So let's do this math again. So 6 EPSPs times 5 millivolts, so 6 times 5 is 30, right? And then we have 1 IPSP of 5, so we can subtract 5 from 30 to have 25. So when we look at our resting potential being negative 70 millivolts, we're going to be subtracting 25 from it to see if we meet the threshold potential. And so when we subtract 25 from 70, we end up with negative 45 millivolts. And if the threshold potential is only negative 50, we have met the threshold and then some, in fact, then additional 5 millivolts. So the answer is yes, you will generate an action potential. So if you were thinking the answer was going to be no just because the previous question was yes, just be careful of that bias when you're answering questions on exams. Don't always think that there's two questions side by side that are similar, that one has to be yes and one has to be no. I just want you to be aware of that.